Alright, so in our previous episode, we discussed basic physiology of skeletal muscle, a muscle that connects to your skeleton. We discussed different levels of organization of the tissue within the muscle, going from the muscle itself, so an organ level, to muscle fascicles, or bundles of muscle fibers or muscle cells. And then we ended by discussing myofibrils. And I said that we really want to understand the mechanism of contraction of the muscle, we need to zoom in a little bit further to look at this thick and thin filament. So in this episode, we'll look at these filaments, learn what they're composed of, and finally discuss sliding filament theory, which is a theory behind muscle contraction. Let's first talk about thick filament. Thick filament is composed of myosin, which is just a protein molecule with a very distinct look. Looks kind of like a golf club. So this golf club has two distinct regions. The first one is called globular head. Another one is called tail. I should also say that the molecule is composed of two identical subunits. You can see two heads there and two intertwined tails. You should also keep in mind that the molecule has two hinge points, one at the tail and another one is at the neck, so right below its head. The molecule is pretty small. It's about 100 nanometers, so you can only see it with one of those light microscopes. Now, if you zoom out just a little bit and imagine this guy is your myosin molecule right here, you can see that staggering these molecules lengthwise and in the regular pattern will give you this thick filament. So the thick filament has two halves or two distinct regions, which are just mirror images of each other. In each region, myosin molecule's tail is pointing to the center of the thick filament, and the heads are protruding outwards from the thick filament. Now, I also marked cross-bridge regions right here. These are the regions on the myosin molecule that will allow it to interact with the thin filament we'll discuss in a second. And they will interact with them through these two binding sites. The first one is called actin binding site, and another one is myosin ATPase or ATP splitting site. Now let's talk about thin filament. Thin filament is primarily composed of actin, but it also has two other protein complexes in it called tropomyosin and troponin. Let's first talk about actin though. So actin molecules are these little spherical molecules with a distinct binding site from the myosin from thick filament. These molecules join together into the strands to form what we call actin helix, that is, a backbone for thin filament. Now, you have probably already guessed that some interaction between thin and thick filaments will lead to muscle contraction. So, let me give you a context. If you want your arm to be relaxed, you obviously want to prevent contraction. Thus, that means you want to prevent anything from binding to this active site on actin, right? So that's why we have tropomyosin. Tropomyosin is a thread-like protein that grooves around actin helix and covers all of the active sites, kind of what I'm showing right here. So then the remaining one is troponin, right? Troponin is composed of 3 polypeptides, just a bunch of amino acids. Each polypeptide has its own role. One binds to actin, another one tropomyosin, and the third one can bind to calcium. Now, troponin, when it binds to actin and tropomyosin, it stabilizes the connection between the two so that tropomyosin does not just randomly slide and expose the active sites. But when the calcium is cycling in the system, it binds to troponin. Troponin pulls onto the tropomyosin and exposes the active site on the helix so that the myosin from the thick filament can bind and muscle can contract. Now that you have all of the background, we can finally discuss sliding filament theory. Now that we get to talk about sliding filament theory, you probably have already guessed that, we discussed two types of filaments and they probably slide to produce contraction just based on the name of the theory. So you may ask, where are they exactly? Like, what's the anatomy of these filaments within the muscle cell or muscle fiber? So people asked that question a while back. They looked at the microscope and this was something they saw. So you can see there are different dark or light region, what we call bands. And the most prominent one there is the dark one called an A band. So the A band, if you zoom in a little bit, you will see thick filaments that I'm showing in red here and thin filaments shown in green. So the A band is the thick filaments and just a little bit of thin filaments in there. Now the I band is a brighter band and it captures only part of thin filaments. So what makes the difference in color is a thick filament. Thick filament makes it a little bit darker. Absence of thick filament makes it a little bit lighter. So then another essential here is this line right here, which I marked in there, called M line. M line is a structure, the protein structure that supports the thick filaments, kind of glues them together. Then another one is the H band. H band is just a space, a middle, the central space of the thick filaments. And then the Z line, you can also notice Z line on that microscopic image. The space in between Z lines is called a sarcomere and is considered to be the smallest contractile unit of the muscle. So what contracts as a muscle? Sarcomere does. Now, a couple of things people usually don't emphasize, but you guys got to know this. There is another protein right here attached to the thick filament called titin. Titin is responsible for producing additional support in a case of muscle strain, and it may be also involved in some of the signaling pathways. Then another fact to emphasize here, if you do a cross-section of the muscle fiber or muscle cell, you will see that the thick filament is always surrounded by 6 thin filaments, and the thin filament is always surrounded by 3 thick filaments. Well, now that you know anatomy, let's actually talk about how contraction works. So in the simplest use case scenario, when you want to contract your skeletal muscles, you first got to think about it. When you think about it, your central nervous system, composed of many little cells called neurons, sends a descending neural command down the axon of your neuron, where axon is just an extension of the neuron. So this descending neural command, also called an axial potential, travels to the end of the axon where it prompts the vesicles to release their contents called neurotransmitters into the presynaptic cleft, where presynaptic cleft is just a gap between plasma membrane of the muscle cells we've been talking about and the central nervous system. So these neurotransmitters cross the membrane through a chemical channel, which causes ionic currents start flowing throughout the membrane. So this current starts flowing, it reaches the T-tubule, which T-tubule can be thought of a structure or really a tunnel leading to a sac with calcium. You guys remember why calcium is important, right? It will bind to troponin, troponin will pull onto tropomyosin to modify its form and expose that actin for myosin to bind to it. So anyways, the ionic current will reach the sac with the calcium that we also call sarcoplasmic reticulum. And then calcium will get released so that troponin will be able to bind it. When troponin binds it, it modifies the shape of tropomyosin. And now you can see that actin binds inside is exposed. So you guys remember that myosin heads have 2 bind inside, actin bind inside, and another one for ATPase. So it binds to the actin, but it also has ATP in the form of ATP and phosphorus already bound to it. So now it uses ATP and phosphorus as an energy to stroke or propel the thick filament along the thin filament, releasing the phosphorus during the contraction or a stroke and ATP after the stroke. So now, when we think about this propelling movement, if we look at our previous picture right here, if myosin heads start contracting or stroking, you will see that this whole region, this whole sarcomere starts shortening. And in terms of muscles, when something shortens, it contracts. Just think of your bicep. If it contracts, it shortens. That's why you see this bump here, right? So then, if there is no excitatory command coming down anymore, your myosin will stop binding to actin, and calcium will be slowly detached from the troponin and then sucked back to sarcoplasmic reticulum through this ionic pump called calcium pump. So this, in a nutshell, was sliding filament theory. I hope you enjoyed the video, and I'll see you in the next one. Thank you for watching.